So I'm here today with Dr. Nadine Ritter, and we are going to be talking a little bit about cell and gene therapies and analytical science in terms of cell and gene therapies. So my first question to you, Nadine, then is what would be your top 3 bits of advice for anyone moving into advanced therapies or cell and gene therapy industry? We're seeing a lot of people moving in from biotech and small molecules. From an analytical perspective, what would be your advice?
So it depends upon where you're coming from going into that field. It's certainly a very exciting field. There's a lot of important therapies that are being developed with it, but the CMC elements of it are very challenging. So if you're coming from you're beginning your career and you're coming into this as a starting point in the industry, then what one piece of advice would be to read on what we've done in the past. Read the guidance documents that have existed for the classic biologics, the current biologics, and then also read what's coming forward for cell therapies and gene therapies, which we tend to lump together in one sentence, but they're in fact very different modalities and have very different process and analytical issues associated with them. But there's a tremendous amount of information available at your fingertips if you know what you're looking for and know where to look and what to read. And I can certainly say that having been in this field now for over 30 years, when I first came in, there wasn't an internet. So everything was sort of learned by hunt and peck and word of mouth and going to meetings. But today, as long as you have enough of a framework of knowledge to be able to place the pieces in context to each other and look at the chronological history, then coming into this field new can be a very interesting way to get started. Coming into it new, I would also say, because I came into it as a PhD protein chemist, I don't underestimate coming into it at the lab level. I came in as a postdoc back in the late 80s and 90s, and I learned that things that happen in QC are amazingly informative and educational, that the basic science knowledge that you have takes you only so far. And once you get into industry, you have to begin to understand the practicalities of it and the constraints of it. And I've had numbers of people that have worked with me, that I've hired myself, and that have gone on to incredibly rewarding careers because they came in through the QC lab level. And at least at that point, you can't make any mistakes because they train you very well to do things very consistently according to plan. And that gives you some time and a good income to be able to find your foot in the field. And cell and gene therapy is right on the cutting edge of that. If you're coming at it from a small molecule background, I would say, don't be scared. It's a very, very different world. We have 250 years of experience with regulations and industry practices for chemical drugs. And so there's very, very well understood science, and there's very well understood regulations and very well understood compliance for small molecules. The same principles apply to biologically derived products and to cell and gene therapy products, but the practices are radically different. And so don't be alarmed. Everything that you know about the principles will translate, although very little about the practices will. But again, it can be very rewarding if you take that experience base and translate it into these modalities. And then the last population of people that are coming in from the normal biologic side, from the existing well-characterized biologic side, or vaccines or plasma derivatives, everything you learned so far will be magnified by what you're doing now. So one of the really fantastic movements I've seen in the cell and gene therapy CMC world is how many of my highly respected, highly experienced regulatory and industry colleagues that's made, major contributions and essentially created a lot of the technologies and regulations for our current biological product therapeutics and vaccines are now taking their seniority over to cell gene therapy products for CMC, and that's going to accelerate the remaining gaps, I think, very quickly. I think the cell and gene therapy CMC space struggled a little bit because it was so novel, and a lot of the people who were working in it and developing it were either medical scientists or basic academic scientists, and that gets you started, of course. But at some point, you need expertise that knows how to bring things into focus for a regulatory perspective, from a quality perspective, and then along with that, good science. And I'm really quite excited, the meetings I've been going to over the last couple of years, to see how many of my colleagues that have done great work in one field are going to make great strides in the other.
Thank you very much, Nadine. And my next question is, and you've probably actually, you have touched on this a little bit actually in the last question, is what do you think the biggest challenges are for people in your sort of role in the analytical world when it comes to cell and gene therapies? What are the big challenges that they face?
Well, the biggest challenge that we faced is how to translate the principles and practices that we've learned with other modalities into these even more squishy modalities. I joke a lot about, we're dealing with squishy methods and squishy molecules, and that's true. And with cell and gene therapy, that's on steroids. The gene therapy world has a lot more consistency and control associated with their modality of product. They're amenable to being stored as a drug substance and being formulated as a drug product and have longer shelf lives. But with the cellular therapy products, and I've worked on many of them. I mean, the sexy one right now is CAR T, but I've worked on a number of cellular therapy products for many years, and they are individually very challenging. And it does take the best science you can bring to the table to try to figure out how to do things correctly to achieve the compliance goals and achieve the control goals, the total control strategy for these modalities. But again, people with experience that are able, I mean, at the beginning of Well Characterized, that's exactly what happened. There were no monoclonal antibodies that were being produced in a recombinant way in the beginning, and very clever people got together and pulled science discussions together to have regulations generated, guidances generated, practices generated, and the field exploded because smart people did their job. And that's the same thing here. Smart people do their job, but it does challenge you to try to wrap your head around, how do I do this validation of that method? What are the principles that apply over here, and how can I make that happen experimentally? That's a fascinating challenge, but we have to do it. As patients, we need the confidence in these products. We need to be confident that they're safe and effective and consistent and stable. And so it's not a challenge we can shirk off.
Thanks, Nadine. And my next question then is, what do you think are the most exciting areas in cell and gene? And that could be, I suppose, therapeutic areas, modalities, interesting technologies. What do you think? Yeah, what excites you at the moment? Things you're working on or things you read about, read about?
Yeah, so I can give you a personal piece of information about the excitement of modalities and emerging therapeutics for things like cancer. I'm getting to the age now where a lot of my friends and family members are having cancer. Fortunately, we have a lot of tools in the tool belt to help with those, if not cure them, certainly buy them a longer life. But I became very much acutely aware recently of the clinical impact of delays in timelines. My younger brother was unfortunately diagnosed with aggressive cancer during COVID. And although he was given 11 months to live, we got him to 22, and he died just 2 weeks before starting a clinical trial with 2 monoclonal antibody products that I've been working on for several years to get them to clinical development. So I'm aware of the need that patients have every day that we delay means that it's a day that somebody doesn't get an opportunity to try the products, right? Even in the clinical space. But what that means to me is that we're all in a hurry. Don't waste time. Don't make mistakes that are avoidable if you only know what to do correctly. So my biggest message to industry, people who are very wanting to go very fast. I've never had a project ever that had a luxurious timeline and infinite amounts of money. But my message is, know what you need to do, when you need to do it, and do it right, and do it once. Because if these patients really are the reason that motivates you to work on your products, let's make sure that we get to them as quickly as possible. But even as FDA said during the COVID vaccine era, we can't cut quality corners because we'll cause more risks than we're benefiting people. So we know what the rules are. We know what to do. Please don't think that they don't apply to you. They do. And just don't make mistakes that cause you to reset the clock. Just go forward with confidence that you know where you're going and what you're doing.
Thanks, Nadine. And off the back of that answer then, so you mentioned that, speeding up the process to get these therapeutics to market to patients. So obviously recently with COVID, we had the fastest ever vaccines made in our history. Maybe correct me if I'm wrong, but it was
Right.
extremely quick. Why can we not do that for other drugs, other biologics, cell and genes, and so on?
That is a fabulous question that I've heard answered incorrectly so many times that I'm very happy to explain that, because even in my own family, they would ask me, well, why can't you do it that fast all the time? Well, there were some very critical circumstances that aligned, that we as a society decided that we would do. One, we decided that we would throw unlimited amounts of money at the effort. So it was literally a space race that we, the public, funded. And so sponsors who received the money were told, don't worry about clinical trials, don't worry about making small scale material, medium scale material, commercial scale material, go straight to commercial. So that collapsed multiple years right there, just the normal time it would take to make material and then run it in clinical trials. The answer was, make it at scale, we'll take the risk that it's going to work. We're not going to use it until we prove it, but don't worry about those intermediate stages. That was massive. That jump started things by years. The second thing that happened is that the decks were cleared. Every CMC person that I knew that was working in that area worked 24/7, 365 days a year, at the exclusion of everything else they were working on. And the same thing happened on the side of the regulators that were working in the vaccine area for reviews and inspections. Their decks were cleared so that they could spend nothing but 24/7, 365, turning these reviews around as fast as possible and doing these inspections in real time as fast as possible. Can we do that again? Oh, and the other thing is that they commandeered all the contract manufacturing, all the raw materials, everything in the supply chain. And so that meant other products that were critically needed, in development, got paused in many cases. So, but the risk and benefit was, we as a society needed these vaccines as quickly as possible. And so it was done. Can we do that again? Yeah, those circumstances are met again, but I don't believe that they will be. Every disease is important to somebody, but as a society, we have very few diseases that have hit us as quickly and as dramatically and as seriously as something like a pandemic did. So I don't want people to misunderstand that, we're not delaying the development of anything. It's just the normal development process where it was given carte blanche with time, resources, and money to get it done as fast as possible. And it happened. Just like the space race, it happened. But it will take something like that again, I think, to get those stars to align. I hope it doesn't, but that is why. And no quality corners were cut. There were some mistakes that occurred, but the checks and balances in the quality system caught it, and nothing went out the door that had any risk to patient safety or health, and the systems did their job. But when you bring in 600 people a month to hire them new, it's going to take a little bit of time for them to get up to speed, even if they are following, written procedures. It's just, we're all humans. And a lot of us were, scared and stressed.
Yeah, it was a fascinating achievement, I think, wasn't it? Obviously making that vaccine. And I don't know if you mentioned as well, there was also the regulators were all just working on.
Oh, they were. The regulators.
Yeah.
I mean, some of the folks that I know that were in the field, I mean, my God, they literally sacrificed their entire personal life to doing their job. They're the gatekeepers to the public, and they stepped up to the challenge and did a heroic job. It's amazing. We had many different industries working, but there's only a couple of people in the world that regulate that review vaccines and inspect vaccines.
Yeah, no, yeah, it was definitely an achievement. And you're right. I think if it has to happen again, it will be because there is another unfortunate disease spreading throughout the world, which won't be good.
No, but I don't want people to think, because I see it all the time. I see it published. Well, we can always, and especially in seminars, telling VPs and investment bankers that, we can do it again that fast. Yeah, are you prepared to do what it took to do that? Because I don't think so.
Yeah, that's a lot of money.
It's an inconceivable amount of money.
Yeah, it's not going to happen. No, that's a good answer. Thank you very much, Nadine. And I think that concludes this interview on cell and gene, and a little bit there about vaccines and speedy development. Thank you very much for your time.